Wait, I lost my coffee. This was fun.
Captivation.
Okay, we're good.
Filming.
Like just when we're ready.
Yeah, it's filming.
It is 10:26.
I lost my faith in God when I lost my daughter to cancer. Beast. I begged, I cried, I offered my life for hers. And day by day, I watched that beautiful little angel slip off. So, excuse me for not taking my seat next to you on Sunday at church. I feel too cheated to worship.
Vince Neil, lead singer of Motley Crue, popular rock band, said this after losing a loved one to cancer. Even though we've been studying pathways and mechanisms of cancer for almost a full semester, we have to remember that cancer is not just proteins and pathways. It's a disease that touches all of our lives. Chronic lymphocytic leukemia, also known as CLL, is cancer of the blood and bone. Mantle cell lymphoma, also known as MCL, is cancer of the blood. Both are B-cell malignancies, meaning cancers of the B lymphocytes, a specific type of white blood cell in the body to fight infection. The 5-year survival rate for CLL is 60.2%. The 5-year survival rate for MCL is 27%. As you can see, up on the board is a picture. What is this? These are white blood cells, and stained in purple are corrupted B lymphocytes, with the arrow next to it.
A drug that can be used to fight B-cell malignancies, such as CLL and MCL, is called Imbruvica, which is commercially sold as Imbruvica. It was developed by Pharmacyclics Incorporated, which is centered in Sunnyvale, California. It was approved by the FDA to treat MCL on November 13, 2013, and to treat CLL on February 12, 2014.
The B-cell receptor pathway is the pathway responsible for the production of B lymphocytes in the body. It is also known as the BCR pathway. BTK, Bruton's tyrosine kinase, is an integral part of this pathway. Up on the board is a diagram of this pathway. BTK phosphorylates itself. Phosphorylate BTK along with SYK phosphorylate PLC gamma 2. PLC gamma 2 activates DAG and IB3. DAG and IB3 activate integrins, which lead to cellular proliferation and production of B lymphocytes. Ibrutinib covalently bonds to BTK, irreversibly inhibiting it, and therefore inhibiting the B-cell receptor pathway, and therefore halting the production of B lymphocytes in the body. In a study published in the New England Journal of Medicine, researchers looked for the effectiveness of ibrutinib as a BTK inhibitor by looking at the amounts of lymphocytes being produced in the body. Up on the screen is a graph showing their research. The purple line, ALC, stands for the absolute lymphocyte count. It's the amount of lymphocytes in the bloodstream. The green line is the size of the lymph nodes. As you can see, at around month 2, the study was over a course of 18 months. At around month 2, there was an increase in the amount of lymphocytes in the bloodstream, as well as a decrease in the size of lymph nodes. What happened was the lymphocytes moved from the lymph node, making it smaller, and into the bloodstream, increasing the amount of lymphocytes in the bloodstream. As you can see, over the course of the 18 months, ibrutinib effectively decreased the amount of B lymphocytes in the body, therefore proving that it effectively inhibits the BCR pathway.
Ibrutinib has also been shown to have potential to fight another type of cancer called T-cell malignancies. These include many different types of lymphoma. This is because it inhibits ITK, or interleukin-2 T-cell kinase, which is a part of the IL-2 interleukin-2 pathway. This pathway occurs when ITK activates phosphorylation, which then activates calcium protein channels. These protein channels let calcium into the cell, and the calcium activates STDK, which inhibits RB. RB would otherwise inhibit E2F, which then activates the cell cycle. Since ibrutinib inhibits ITK, ibrutinib therefore inhibits the cell cycle. On graph D, we can see how effectively ibrutinib binds to ITK. The x-axis represents whether we're measuring pre-dosage or post-dosage. The y-axis represents the percent of ITK in the body that is covalently bound by ibrutinib. As you can see, before dosage, no ibrutinib was bound to ITK, and after dosage, every patient, represented by a dot, had at least some ITK bound by ibrutinib, proving ibrutinib an effective ITK inhibitor, or ITK binder. Graph G represents how many T cells are in the body before being given ibrutinib and after being given ibrutinib. The y-axis represents the number of T cells in the body, and each of these lines represents the average amount of T cells in the body over several patients. Yes?
Is pre-dosage before ibrutinib was administered to the body?
That's correct. As we can see, pre-dosage, there was more T cells on average than post-dosage, showing that ibrutinib effectively decreases the number of T cells in the body, therefore decreasing the danger of the cancer. Another therapy that is often used to treat B-cell malignancies is called chemoimmunotherapy, a combination of chemotherapy and immunotherapy. Chemotherapy is used to stop the survival and growth of cancer cells, whereas immunotherapy is used to restore the immune system, since killing the cancer cells may also kill healthy immune system cells. Chemoimmunotherapy has been shown to be very effective for younger patients. However, it's not quite as effective, and there are serious side effects when given to the elderly. On the other hand, ibrutinib is very effective on the elderly, but is not approved for use on children. Since B-cell malignancies are more common in the elderly, in this sense, ibrutinib is a more effective drug. However, chemoimmunotherapy does have a mechanism to restore the immune system, whereas ibrutinib doesn't, which is going to make patients more susceptible to illnesses. In this sense, chemoimmunotherapy is more effective. Furthermore, people have been shown to develop a tolerance to ibrutinib, therefore making it less effective over time.
As an effective BTK and ITK inhibitor, and therefore an inhibitor of the BCR and IL-2 pathways, ibrutinib is an effective therapy option for B-cell malignancies, including MCL and CLL, as well as T-cell malignancies. Do you have any questions? Yes.
Okay, so I know that we were talking about T cells and B cells. Okay, so what is that connection between the T cells and B cells? With the BTK inhibitors?
Both are types of white blood cells.
Okay.
And they both are parts of the immune system. They fight against infections. Does that answer your question?
Yeah, and so you're targeting the immune pathway.
Not
Yeah.
You talked about how BTK phosphorylates itself at the beginning of the presentation.
Yes.
What stops BTK from phosphorylating itself too much? Can BTK phosphorylate itself too much? I mean, I feel like if it can just phosphorylate itself, what's keeping it from phosphorylating itself all the time?
I don't know.
Are there any other questions? We'd like to thank Dr. David Cooper, Dr. Michelle Meyer, Dr. Norman Woolmark, Luke D., and Mark Stroffman for their help with this project.
Good job, guys.
So how are B cells and T cells specifically targeted?
Because we're targeting the pathogens of the production. We're not targeting the ones that are already existing. We're targeting the production of those.
Right, but you said that BTK targets E2F.
E2F is a part of the pathway that it targets.
Ibrutinib through ILK,
ITK.
targets E2F.
Yes.
Can you go to the next slide?
What's the T cells and B cells?
So, because it's targeting ITK, if ITK is being inhibited, then this whole pathway is inhibited, and therefore E2F is not going to run. Does that answer your question?
Right, but that would make it non-B cell specific. E2F is present in everything, right? In all cells.
Yeah.
So how is this specific? And what you're showing in your data is that it's specific to immune cells, correct?
Yes.
Correct.
The-
So how is ibrutinib targeting those cells?
I'm not
Targeting which cells?
The immune cells. If ibrutinib is an ITK inhibitor, and which is upstream of E2F. Does everybody understand what I'm asking?
Why do you go to ITK?
No. So if it inhibits E2F,
Isn't it inhibiting all
is it inhibiting all
cells?
cell cycles? All cell cycles.
What
All
cells?
cell cycles.
I think that since ITK is only present in T cells, since it's inhibiting only E2F that's activated by ITK, it's then only inhibiting T cells.
Yeah.
How is it targeting both the ITK and the BTK? Like, is this because I know that you were mentioning different types of cancers at the beginning of the presentation, so are these like how they target the different types of cancers? Or
So-
is it like,
Both mantle cell lymphoma and chronic lymphocytic leukemia are B cell malignancies, so that's the BCR pathway. And T cell malignancies are from the IL-2 pathway. And the reason that it binds to both of them is because they're structurally similar, and that ITK is downstream of BTK, like in the process. And so it just binds to both.
so why would they be targeting the T cells if it's a B cell malignancy?
It just presents or it is able to
ITK and BTK have very similar structures, so it is capable of inhibiting both of them because it bonds to both of them.
But why would you be inhibiting both B cells and T cells?
I'm not sure I understand the question.
Because the chronic cell is a chronic
Chronic lymphocyte.
yeah, and so your two like the two cancers that are actually B cell malignancies. So why would you be targeting a T
This
cell?
It
is
just happens-
showing
it
that
also-
this is showing potential for it to also be effective in a different type of cancer.
Okay.
This isn't relevant to B cells and CLL and MCL.
Okay, so this is like showing promise for the future.
Right.
Okay.
Does it do anything about B cells that are already existing? So what happens to those?
well, I mean, I don't specifically know the lifespan of the lymphocyte, but it just halts the production. The drug is just ends up inhibiting the pathways of production.
How long after dosage was that?
I'm not sure. Are there any other questions? Sean?
What's the administration cycle for ibrutinib?
Orally. Well, it's given orally and in yearly dosages.
Not exactly.
It varies on the study. I mean, in a study in blood, it was shown there has to be at least 2.5 milligrams per kilogram, right? Or milligrams per kilogram given a day for it to be effective.
Okay.
So,
it's a
I
daily
mean,
drug?
excuse me?
It's a daily drug?
Yes.
Okay.
Any other questions? Thank you for your time.
Oh wait, we have to leave the room. Yeah.
Can you cut the video, please?
Oh.